Welcome to the MEF2C cast, the podcast advocating for the MEF2C haploinsufficiency community by broadcasting the voices within. I'm Eli.
And I'm Haley. We are parents to a boy with MCHS and autism, sharing our journey with you throughout conversations grounded in learning, support, and community.
This is Advocacy Amplified. Do you have questions, comments, or a story to share? Want to be a guest on the show? Connect with the MEF2Ccast on Facebook, mef2ccast.com, or email us at mef2cast@gmail.com.
If you enjoy the show, please support us by subscribing to the show on Apple and Spotify, leaving us a review, and following us on social media. You can also buy MEF2Ccast merch at our Shopify store, linked in the podcast description. The MEF2Ccast is for informational and entertainment purposes only. Views expressed are those of the hosts and guests and not necessarily of the podcast or its sponsors. This is not medical advice. Consult a healthcare professional for medical concerns.
Alrighty, hi everybody. Welcome to the MEF2Ccast. Eli here, hopping on to give a few quick announcements before our interview. First of all, our interview this week is with Dr. Christopher Cowan. During the interview, we had a variety of issues. Chief among them was there was a terrible windstorm that blew through during our interview, so our internet connection went out part of the way through, and then there was some other internet weirdness and glitchiness and stuff, so we're hoping it pieces together okay. We'll do our best to make it as seamless as possible and avoid any technical weirdness or anything like that. But if there's any odd sort of vibe to the podcast, that may be why. The other announcement we have is not related to the podcast. So the U.S. MEF2C Foundation, who we have no affiliation with other than we think they do wonderful work, we think all the foundations do, is hosting a meetup in the Cincinnati, Ohio area, arriving the evening of June 6th. Group activities that evening, group activities all day, Saturday the 7th, and then maybe a group activity in the morning and leaving on Sunday the 8th. So if you guys have never been to meet another family with a child or, whoever with MCHS, I highly recommend it if you have the money and the time to come out. Haley and I were fortunate enough to go to the conference in Boston last year, guys, and we'd never met any of these people, and we'd only had our diagnosis for a few months. And walking into a room full of these parents and kids, I can really only describe it as going home, but to a home you never knew you had. That's the only way it feels. There's such a sense of family and just belonging, and it's just incredible to observe all these people be themselves. So even those words that I've just spoken can't truly describe how it feels, but if you can, please come out. We'll be there. Michelle, who is organizing the whole thing, shout out to Michelle, thank you so much, is planning a group trip to the aquarium, so there will be costs associated with getting into the aquarium, but she's got us discounted tickets. She's got a block of hotel rooms at a discount for us, so check out the Facebook group to learn more. And please come out, guys. Like I said, we're coming, so we'd really love to see you there. So now I'll go ahead and introduce our guest, Dr. Christopher Cowan. Dr. Christopher Cowan is professor and chair of the Department of Neuroscience at the Medical University of South Carolina in Charleston. He also holds the Smart State Endowed Chair in Brain Imaging. Dr. Cowan is also the director of the NIH-funded Center of Biomedical Research Excellence on Neurodevelopment and its Disorders, and the co-director of the new MUSC Neuroscience and Brain Health Institute. He is also the co-founder of Neuroepigenics, LLC, a company advancing a novel therapeutic treatment for alcohol use disorder. Dr. Cowan earned his BA from Wesleyan University and his PhD in cell and molecular biology from Baylor College of Medicine. He completed his postdoctoral training with Dr. Michael E. Greenberg, who I might add is also doing some great stuff around MEF2C, at Harvard Medical School and Boston Children's Hospital in the area of molecular neurobiology and brain development. Dr. Cowan began his independent research lab as an assistant professor in the Department of Psychiatry at the University of Texas Southwestern Medical School. In 2012, he became an associate professor of psychiatry at Harvard Medical School and McLean Hospital before being recruited to the Medical University of South Carolina in 2016. Dr. Cowan's NIH-funded research program focuses on genes and molecules controlling the development and wiring of the brain, including genes linked to syndromic forms of autism spectrum disorder like MEF2C. He has been studying the role and regulation of MEF2 genes in typical brain development for more than 25 years. During the last decade, his research group has developed animal models of MEF2C haploinsufficiency syndrome and gained critical insights into how reduced MEF2C function impacts brain development and neurotypical brain functions. His group has spearheaded the development of several innovative therapeutic strategies to potentially treat MCHS. So, guys, please enjoy our interview with Dr. Christopher Cowan. All righty, welcome to the MEF2C cast. We are here with Dr. Christopher Cowan from the Medical University of South Carolina. So, Dr. Cowan, can you start by telling us a little bit how you got involved with MCHS?
Sure. Yeah, I'd be happy to. And thank you for hosting me for this. This is a great idea. I got interested in this many years ago, studying the gene MEF2C and its close family members, MEF2A and MEF2D, and really been studying MEF2 proteins for going on 20 years. I originally got interested in this from the standpoint of trying to understand how the brain typically develops. And at the time, we were using rats and mice to study, a model system of brain development. And genes like MEF2 family members came out as interesting candidates to explore. They were originally cloned, originally identified as being highly expressed in developing muscle and developing brain. It's two separate groups. And so we started looking at what MEF2 proteins might be doing. It was particularly interesting because as the top parts of our brain, the cortex, begin to develop, as that part starts to expand, MEF2C protein in that region really starts to express at high levels. It starts to be present at very high levels. And we really wanted to understand what it might be doing. And then the other part that was interesting is that during brain development, neurons, which are the, excitatory cells in our brain that form connections, and it's part of the network, right, by which information is received and processed and sent out to our bodies to respond to sensory and experience information that we have, that we experience in our lives. And during our brain development, there are factors like MEF2C that play a role in telling the neurons how and where and when to form those connections. And importantly, they also play a role in a process we'll touch on a little bit later, I suspect, in how those connections are remodeled, right? Because initially it's kind of a crude map. And through our life experience, our brains begin to prune away those that are inappropriate and strengthen and keep those that are appropriate. I got interested in MEF2C for a couple of really kind of seemingly trivial reasons. One was I was just doing an analysis of all the three major MEF2 proteins, A, C, and D, which are all very, very similar to each other, and found that when we knocked out MEF2C, it had the most profound effect on brain development. And that made me think that this gene was probably super important. And so we began focusing a lot of effort on that. And then in about 2010, the first clinical literature paper started coming out. Actually, the very first one, which reported, I think it was 13 patients. Most of them, 11, I think, were deletions, and 2 of them were point mutations. And they were in different sizes of deletions. And when they lined them up, everything lined up on MEF2C. And the patients had very similar developmental disorder symptoms. And so this led that group to conclude that MEF2C, that deletions in one of the MEF2C genes probably causes a disorder which we call MCHS now, or MEF2C haploinsufficiency syndrome.
Um, excuse me. So piggybacking off of that, can you give us a very brief overview of MCHS, both in terms of the gene itself and then, well, you've already explained a little bit, but the role the protein plays in the body?
Yeah, sure. So MEF2C is actually a very big gene. It's one of the longest genes we have in our cells. And this gene, when it's made in the cell, when it's converted from the gene to the messenger RNA to the protein, it becomes what we call a transcription factor. So this is a type of protein that functions in your cell nucleus to express other genes. So I like to sort of think of it as it's like the photocopier function in your DNA. So your DNA, we have greater than 20,000 protein coding genes, and a neuron expresses greater than 13,000 of those. So not all genes are expressed in every cell. MEF2C tends to show a restricted pattern in certain types of cells, like I mentioned, skeletal muscle, cardiac cells, cardiac muscle cells, some immune populations, as well as in many populations of neurons in the brain. So when this protein is created, it is then recruited to other genes by which it makes copies of those genes. So I like to think of this again. It's sort of like your genome, the DNA in your nucleus is like the blueprint, and the transcription factors come in, and they basically, they're like the foreman on the construction job. They tell which of the genes need to be turned on in that cell, how much, and then those photocopies, which we call the messenger RNA, those are the instructions for the cell to then convert to protein. And genes like MEF2C are super important, all these transcription factors, because of this function, right? They are able to regulate hundreds or thousands of other genes in our cell nucleus. And so you can imagine that loss or reduction or altered function of a transcription factor is going to have a cascading effect, not just on one thing, but the hundreds or maybe thousands of different downstream things that are affected.
That makes sense. I've compared it to if you imagine each cell is a piece of IKEA furniture and the machine at the factory misprinted the directions. So, you're starting at step 2 and you misbuild the piece at step 2. And then because you misbuild that piece at step 2, it doesn't fit right with this other piece. And as you go downstream, there's all these, as you said, cascading effects.
Yeah, absolutely. I think that's a great analogy.
I appreciate that. So how much of MEF2C's function is during uh, development? And sort of, is there a window where it's more crucial for neural development?
Yeah, that's a great question. We don't have a complete answer to that yet. We do know that MEF2C begins to be expressed very early in brain development. It seems to be a factor that tells particularly the cortical part of your brain, that outer, gyrated lumpy part that we think of when we think of a human brain. The neurons within that population are becoming neurons because of MEF2C coming on. And because of that, it is playing a critical role in the formation of the early brain. So we know that from studies in animal models in particular, we know that MEF2C plays a role in different ways in which the neurons are being what we call differentiated, or basically specified into subtypes of neurons, because the brain isn't all just one type of neuron. There are hundreds of different subtypes of neurons. Some of them are excitatory, meaning that when they're activated, they activate a downstream cell or muscle or whatever effector cell population. And some of them are inhibitory. When they're activated, they put the brakes so that the downstream cells don't react to information that may be flowing. And so actually it turns out MEF2C is expressed in both excitatory neurons and inhibitory neurons, and is important for both. And this is also why it's really complicated to answer your question, because it's playing a role in both of these cells, and how the complex network forms in our brain and the way that information is being processed is super complicated. We have billions and billions of neurons in our brain forming trillions and trillions of synapses, which is those sites of connection. So you can imagine trying to solve that puzzle is incredibly challenging, but also highlighting the fact that something like MEF2C that is at the heart of the formation of these types of cells and how they form their connections, it's not surprising that it turns out to be really critical for neurotypical brain function.
So my understanding is not only at the neural level with the excitatory and inhibitory neurons, but at a cellular level, MEF2C can act as a repressor and activator too. Can you explain that a little bit?
Yeah, that's a really interesting phenomenon. So a lot of transcription factors. So if you think of the transcription factors, as a blob, right? So the different parts of the blob are doing different things. So one part of the blob of MEF2C and other transcription factors is really designed to bind to your genomic DNA. That's how it sits on the right gene and turns it on. Another part of the gene of the blob is a part that recruits other blobs that help it to turn genes on or to turn genes off. So that's the whole idea of, like, if it if it binds together with a co-activator, we call it, that helps to upregulate that gene and increase its expression. If it happens to be binding with what we call a co-repressor, then that If that thing is bound with the MEF2C, it's going to repress those genes. Now, to be frank, we don't yet understand the code. We don't know why sometimes MEF2C acts as an activator and sometimes acts as a repressor. We do know a little bit. So a number of years ago, my group and others discovered a mechanism by which when a neuron is very active, it switches MEF2C from an inactive repressor type form to an activator form. And that gives the cell the capacity to be dynamic, to respond to changing information. So as cells are getting sensory information and they're processing, MEF2 can be switched by the cell from the repressor to the activator to drive a program of gene expression that presumably does something to the cell to help it, form and adjust.
That's really interesting. Can you explain a little bit about the relationship between autism and MCHS? Is MCHS a type of syndromic autism?
Yeah, so MEF2C is in the Simons Foundation Autism Research Initiative Safari gene classified as an S1. So the one means it's the highest confidence of risk for autisms, meaning it's a gene that they feel very confident saying this is an autism risk gene. The S indicates that it's syndromic, and the syndrome just means that in addition to the core symptoms of autism, so remember, autism is defined by repetitive and restricted patterns of interest and activities, as well as social communication and social related challenges for individuals. But those are just the core symptoms. If you have other symptoms layered on top of that, right, which sort of we could casually call this like autism plus, right? There are other common symptoms associated with autism. And in the case of MCHS, that's definitely the case. We have seizures that are extremely common in upwards of 80 to 90% of individuals, often before the age of 2, although in some cases a little later. Usually motor coordination problems. Many individuals have challenges with intellectual, cognitive function, intellectual disability, and a number of others that are commonly seen. We did the first in the world natural history study of 73 individuals with MCHS. And it was interesting to really see the relative penetrance, this is the jargony term I'll use, but how frequent we would see certain symptoms with individuals, and seizures were right up there on top. Absence of speech or limited speech is way up there in terms of the relative percentage, et cetera. So anyway, that's a long way of saying that MEF2C is an autism associated gene, but it's autism plus these other common co-occurring symptoms.
So is autism a category in which MCHS sits? Is that the right way to think of it? Or are they things that just tend to occur together often?
I would say that autism is there aren't as many examples of individuals that I'm aware of that have MCHS or that have MEF2C mutations that don't have features of autism. Now, to clarify that, if you actually looked and tried to document the fraction or percentage of patients with an autism diagnosis,
Mm,
it's not 100%.
right.
However, in many cases, that's because it hasn't really been explicitly tested.
Mm,
Social communication, social interaction problems, that's probably almost 100% of the population of MCHS has challenges with communication. In fact, many have no words at all in their lexicon. The repetitive, restricted patterns of behavior, et cetera, very, very common to have repetitive motor patterns, hand wringing, and many, many different flavors of that. So if we were to ask every individual with with MCHS, if you were really to do an autism diagnosis, you'd probably find that it is almost 100%.
right. Yes. Gotcha. Okay. That makes sense.
But I should also just add that because it's so severe,
Mm,
in the jargon in the field of autism, we would call this profound autism.
right. Yes.
So I think that that's the way to think of it. MEF2C is a profound form of autism, but because of the cluster of other symptoms, it really is also a sort of almost a separate thing that overlaps with autism.
I see. Gotcha. Yeah. See, since we've got our diagnosis for whatever reason, I'm very interested in kind of the taxonomy of how it works. So, and like I said, to be honest, I'm not sure why it interests me so much. But anyway, can you talk a little more about MEF2C's role in the brain? with that neuronal pruning and that the excitatory inhibitory imbalance and sort of how that manifests in the symptoms that we see?
So a number of years ago, we discovered that MEF2 proteins, and originally we were actually studying MEF2A and MEF2D, but we find that MEF2C does something very similar. When MEF2 proteins are activated, if we increase their function, they do something really interesting in the developing brain. They seem to prune or eliminate some of the synapses that are being formed. And it's not all of them, it's just a subset. And this was a really exciting discovery we made more than 20 years ago now that MEF2 proteins are sitting in this really interesting spot where as the neurons are getting more and more connected as the brain is developing, and those because of those connections, those neurons are becoming more and more active. The cell, we think, has evolved this feedback mechanism, right? Mother Nature has provided the cell a mechanism to say, okay, we're super active here. We need to dampen the amount of activity that's coming in. And we think that MEF2 proteins are sitting at that intersection. They're able to drive a program that tries to prune away some of the inappropriate synapses and help sculpt the final mature circuitry within our brains.
So that would be like why maybe people with MEF2C have a hard time integrating reflexes, for example, like when they're babies and the reflexes don't go away.
I think it's probably at the core of a number of symptoms. It's probably at the core of motor coordination.
Mm,
It's almost certainly at the core of sensory information processing.
mm,
Many of the individuals with MCHS have either increased or decreased sensitivity to
right.
different sensory stimuli. It may or may not be the case that their primary sensory out on the periphery, that they might be able to detect it normally, but how that information is processed may be functioning incorrectly. Too many connections. It's creating noise, so to speak, in the system that makes it difficult to process. And in some cases, that may lead to hypersensitivity
Mm.
stimuli. And in some cases, it may lead to reduced sensitivity because of compensatory mechanisms in the brain that are trying to counteract that. The other thing that I think is interesting to think about also is its relationship to seizures and epilepsy, right? So this is where the EI imbalance piece probably comes into play. If you are not appropriately pruning inappropriate excitatory synapses, you may end up with a shift in that excitatory inhibitory balance. And this has been shown in animal models to be the case. And if you have so if you have too many excitatory and not enough inhibitory, then you can imagine a network which can get recurrently active without being able to stop itself effectively.
Do you think that that might be why so many people have issues with sleep as well?
MEF2C seems to be playing an important role in coordinating the way that and a lot of this is still magic and mystery. I'll be honest. People have been studying sleep function forever to try to understand what happens. When we go to sleep, it's not like our brains are less active. In fact, in some ways, they're more active. But what they do when we sleep is they become coordinated. So we have these waves of coordinated activity in our brain that happens during different phases of sleep. And MEF2C, when you delete it or reduce one allele, one copy of the gene, it changes the way the brain is synchronously controlling activity in the brain. And that almost certainly is due in part to its ability to modulate or, change the excitatory inhibitory function. But a lot of that detail we still need to figure out and look into. But we clearly think that in the MEF2C heterozygous mutant mice, for example, we published a paper a few years ago, and a colleague of mine at UT Southwestern, Robbie Green, that showed that those animals have abnormal slow wave sleep, which is a type of synchronous activity that happens during a particular phase of sleep, and that animals are showing evidence of increasing sleep need, which is something that happens if you're not getting appropriate sleep function. Your brain starts to develop this need for more sleep. I think that particularly for kids that wake up at random times in the night, which is very common in MCHS, it's almost certainly an inability for the brain to maintain those sort of normal patterns of normal sleep function.
That makes sense. It seems like there's, from what I understand, there's some relationship between F2C and dementia. Do you think that there could be? I know the data on adults with MCHS is extremely limited, but do you think there's a potential for increased risk of dementia for adults?
Yeah, interesting. So one of this was originally observed by a group from MIT that showed that in aging mice and in human postmortem sample brains, MEF2C levels begin to decline as we age. So it's really interesting. When we're developing, it skyrockets up as the brain develops, MEF2C levels. And then as we're maturing up to, our brains continue to develop until about 18 years of age or a little later, until things are firmly, fully developed. But then the levels begin to decline to maybe, and at least in rodents, we know it's about half of what the peak levels are in early development. And we maintain those levels through the rest of our lives. And in animals, they notice that animals that had a greater decline in F2C levels showed reduced learning and memory function, so sort of a measure of sort of cognitive function. It's certainly possible, and I think you're right on the money there, that we don't have enough data of
Mhm,
older individuals with F2C haploinsufficiency to know yet what that
mhm,
means. But I'll also add that many of the individuals are starting with severe, in many cases, severe intellectual disability. So it's possible, but it's hard.
Do you think that, well, so there are examples of at least one adult that we know of who can spell with MCHS, graduated with a high school diploma. And there's another guy we know about who can speak, has part-time job. Do you think that how much of it is actual intellectual disability and how much of it may be like information processing and just sort of their ability, like expressive communication?
Yeah, I think you raise a really great point here that I should emphasize, which is that all of us have different. Yeah, we all these individuals may have different mutations and deletions, and I know we'll
Right,
get
yes.
in a little bit. But on top of that, you layer that complexity with the fact that your genes and my genes and anybody out there, we're all different, right? We
Mhm,
inherit different variants from our parents, and that unique combination is our cells then process that and determine how our MEF2C will be, how the effects of it will be expressed.
mhm,
So for in some individuals, they may express other genes at higher levels that compensate for reduced MEF2C levels. And so for
mhm,
those individuals, they're sort of protected. And I think of this also like with cancer, we know this for cancer risk. Some people can smoke their entire lives, 2 packs a day or more, and never develop cancer. Those individuals often have a genetic makeup so that they're able to repair effectively damage that's caused by smoke. Whereas
mhm.
another individual may have gene variants that make them highly susceptible, and they don't, they may develop cancer earlier in life or something along those lines. The same thing goes on for brain development. Our unique genetic modifiers, which is how we think about it, our other genes modify the severity of an insult on a particular gene.
Mhm.
For those individuals, it may be the case that they have other genes that help buffer, that help compensate for the MEF2C mutation. And in other individuals, they may be more vulnerable to the most severe effects, if you will.
Gotcha. Maybe you answered my question and I didn't quite understand it, but I wasn't talking so much about the variance of intelligence in between individuals as like maybe their ability to express their intelligence. So like the guy I was talking about who can spell Andrew, like he and I email each other back and forth. Like once he was able to communicate through spelling, he's of relatively normal cognitive function. So I'm just wondering.
And they didn't know up until that point.
Right. So I'm
He was
just wondering
13 when
how
they figured it
much
out.
of the perceived intellectual disability in general may be due to motor function impair and their ability to express
Express it.
themselves.
Yeah, I think that's a really great point. And that's another sort of difficult part about human cognitive function, right? I mean,
Right.
we're all on a spectrum, right? And you're right. There's certain underlying abilities may be masked by an overriding physical impediment, for
Right.
example, or emotional or other aspects that may override our ability to sort of see what the true fundamental capacity. That's a
Gotcha.
really great point. And in fact, in autism, as you know, a lot of individuals, what they found particularly for some nonverbal individuals with autism, right, that touch pads and things like this
Mhm,
can really help overcome
mhm.
a communication deficit by using other modes of expressing the
Sure.
feelings and thoughts they have.
Yeah. Can you talk a little bit about MEF2C's role in the immune system and the microglia?
Yeah, I mean, I'm not an expert in peripheral immune systems, so
Right.
I don't want to speak authoritatively around here. I know a lot more about its function in microglia in the
Uh,
brain. So the immune system, obviously, these are cells that are born in the peripheral system. They're born from our bone marrow and are produced and constantly being sent around our bloodstream. And they help us to fight, to detect self from non-self, right? To virus infections, bacterial infections, things like this. And for cells that are dying, right? The immune system helps us to prune those and clear them from the system. Well, the brain has a population of immune cells, actually have a lot of different populations of immune cells that can infiltrate. But the resident immune cell of the brain are something called microglia. And these microglia are like macrophages. And so macrophages in the periphery are immune cells that engulf, again, viruses, bacteria, dying cells, et cetera. They help engulf them, break them down, and clear them from our system. The microglia in our brain are doing something similar. This is something we've really only known for the past couple of decades, actually. But these microglia help us to sculpt the connections in our brain. So synapses are being pruned away by the microglia. I sort of think of them as the garbage collector, kind of roaming around, sensing the brain. And as synapses get tagged for, okay, this is one we need to get rid of, the microglia come and clear it. Okay. Turns out that MEF2C is expressed as high, if not higher, in microglia as it is in the neurons in our brain. And it's playing a super important role in those cells, particularly in their development and maturation to become, mature microglia. We haven't published a lot of this work yet, but we have got really great evidence now that shows that, at least in the mouse model of MCHS, that the microglia are showing pretty profound deficits in their ability to prune synapses in the developing brain, at least of the mouse, which suggests to us that MEF2C's function in the microglia is probably going to be playing an important role, at least in part, in some of the brain development dysfunction that may be happening.
Right, that all makes sense.
Yep.
Can you sort of take us through where we're at with TSBs right now, kind of how they work and where you're at in the process of getting them into actual patients?
Yeah, so we've made really surprisingly rapid progress on this, I must confess. It's been really gratifying to see something go from kind of a basic idea of like, ooh, this might work, this would be cool to try, to seeing that it is working. So where we are right now, the whole concept of a TSB. So TSB stands for target site blocker. And without getting too much into the weeds, the whole idea behind this is it's a small modified ribonucleic acid, RNA. And it's modified chemically so that when you inject it into the system, it is not degraded rapidly by enzymes that are naturally present. Okay, so when we naturally make RNAs in our cells, they're around for a short while, they make proteins or they do other kinds of things, and then they're turned over. These modified RNAs, which we didn't discover, we're really leveraging some of the biotechnology that's existing out there already, are being utilized right now for antisense oligo, or ASO may be the term you're most familiar with. But the ASOs are designed to be sort of an RNA and DNA hybrid structure, which is designed to go and tear down and degrade mutant forms of RNA, messenger RNA. So for example, if you had a gain of function mutation in a gene and that's what's causing the disorder, you want to target that for degradation. But in our case, it looks like in almost all cases we've studied, the MEF2C family of mutations seem to be loss of function, meaning that whether it's a big deletion or a point mutation or a frame shift that causes a premature stop, or what we call a nonsense mutation, which is basically like a stop codon, which is kind of like putting a period in the middle of the sentence, that those all seem to be loss of function. So because of that, we kind of took the simple idea that we know the problem. The problem is there are two gene copies of MEF2C, and one of them is not functioning properly. So what happens if we could just increase the function of the healthy non-mutant copy? And so the TSB is designed to interact with the messenger RNA and stabilize it so that we can get more protein made out of that same amount of messenger RNA. So we have designed a number of these. In fact, we have, I think, 8 or 9 now that we've tested in human cells in culture, which just means in a dish in our laboratory, as well as we've tested several of these in mice, injecting them into the brain of the mice. And we're able to see increases of expression anywhere from, let's say, 30% of increase to as much as a twofold, 100% increase. Now, if you think about it, you have 2 copies, you lost 50%. Ideally, we'd like to kind of double the expression of that 50% to maybe have the levels that you would expect with two functioning copies. Now, you don't probably need that much. We don't know what the effective threshold, like when do you have enough to be able for the cell to be able to do what it needs to do? So it may not be twofold. It might be we need to increase it 30% or 40%, right? That might be enough. The other thing is the human brain and the body is just really phenomenal. It has the ability to make adjustments on its own. So in our mouse model, where we delete one copy of the MEF2C, we see that the other copy upregulates its expression about 10 or 15%. So already the brain is trying to increase the levels. So we may just need to give it a little bit more of a push. So where we are right now is that it looks like it's working very well in cultured cells, in both human and mouse cells, and it's working in the mice. And we're in the process of now testing it in the mice to see if it can rescue the symptoms, if you will, in the mice in our mouse model of MCHS.
Are there so the different TSBs that you're testing, are there different use cases for different TSBs, or are you just trying to see what is most effective?
Right now, we're just trying to pick the ones that work the best, meaning they give us the biggest increase. The design of this is really interesting and gets into the weeds too much, I suspect. But the design is it's to interfere with a naturally occurring process in our cells that is designed to limit the expression, the protein production. Okay. So the TSBs are trying to interfere with that process. But cell to cell can be quite different. Different cell types can have different factors that interact and limit the expression. So, for example, TSB number one might work best in an excitatory neuron population, but work not quite as well in inhibitory or in microglia. TSB number two may work really well in microglia and peripheral immune system, but really not well at all in neurons. So we have to work all that out, which is part of the reason why we've been developing so many in parallel so that we can figure out which ones give us the best, cocktail of effects. In the end, it's probably not ideal. So from FDA approval standpoint, every single TSB is going to have to be characterized as a separate drug. Okay. So if we did a cocktail of TSBs, which may actually be kind of a nice formula for getting different cell types to upregulate, we're going to have to characterize each of those as an individual drug, which slows down the process. So our hope is that maybe we can identify a TSB that is overlapping so that in every cell type, it may not work quite as well in every single cell, but it gives us an increase in most cells that express MEF2C.
That makes a lot of sense. You would just have one TSB that you'd be able to give instead of like so much of this one and so much of this one, and I see.
Ideally, again, just because of the timeline for approval
Right.
testing in humans. Now, in
Yes.
the end, it may turn out that a cocktail, and all, multiple
Mhm.
things are characterized, that becomes the best formulation. That is the best strategy. We're trying to sort of balance the urgency to move this forward with what is sort of the ideal perfect world, right?
Right. Yeah. Because MEF2C plays such a big role in development, is there like an age window in which treatment may be more effective or, I don't know exactly how to phrase that, but you get what I'm driving at.
Yeah. So the short answer is we don't yet know. And it's part of the things we're trying to test now. So if we give it at birth, if we give it several weeks later, if we give it several months later, when is the window in which it has effectiveness? My hope is that the window is quite wide. Now, we know in humans at birth, those first 2 years, there's a lot going on in the brains, developing like crazy, right? Dividing, forming connections, and we can see this even in a baby, right? A baby is able to suddenly over a 2-year period develop language, roll over, have coordinated things, begin to speak. I mean, there's a lot of magical things that are happening in a baby around that first 3 years of life. So it wouldn't be surprising that if we can intervene early, that's going to have the biggest impact.
Mhm.
But as I mentioned earlier, the human brain continues to develop and refine and remodel, and MEF2C continues to be expressed throughout our lifetimes. And we know that the adolescent brain is continuing to develop all the way up until 18 plus. So my guess is that for things like language development, it will have a bigger impact earlier in life. And for older kids, it will have less of an impact. But maybe combined with some behavioral therapy, right? Speech therapy combined with this kind of treatment, you can develop these skills later in life. It may be harder. I'd sort of imagine like, if your child is trying to learn 2 or 3 foreign languages at, the age of 2 to 5, that is so much easier for them than trying to pick it up when they're 20, 30 years old. So I really sort of think about it in the same idea there, that are in the same light. The younger is the easier the brains, the more plastic the brain, the easier it is to adjust.
Are there any non-TSB treatments that you're currently working on and you think show promise?
Yes, actually, are also working on a virus-based therapeutic, again, with this idea that if we just need to increase expression of functional non-mutant MEF2C, perhaps one way we can do that is deliver the MEF2C to the neurons in the brain. And again, as one of my students was just describing the other day, she said she's Nigerian, and she said, if you give me the ingredients, I can make Nigerian food. If you give me the type, I can make all different kinds of Nigerian food. We feel the same way about MEF2C, right? We know that if we just give the cell enough MEF2C, it will know what to do with it, and will hopefully have ability to sort of remodel and increase function, and perhaps have really profound increases on quality of life for some of the individuals with MCHS. So we're using something called an adeno-associated virus, and there are more than 170 clinical trials going on in the US right now using adeno's AAV, we call it for short, to treat a whole variety of genetically linked disorders. So what we're trying to do is we're trying to take this AAV. We've got it now expressing MEF2C, and we had to do a little bit of engineering so that it could express the right forms that we need, because it's complicated molecule. But we have this working now. It looks awesome in mice. We're able to get a very high percentage of the neurons in the brain to express MEF2C that should be expressing it. And we are actively testing now to see if we're able to rescue some of the symptoms, the phenotypes in the mouse model. If that's successful, we're already beginning to move forward with, talking to some industry partners to potentially start pushing this forward into potential clinical use. There's a lot of work that has to go into this. The advantage of the virus approach, which is exciting, is that it's potentially a one-shot treatment. These viruses express the MEF2C for decades, whereas the TSB is more like a pill that you have to take, right? It has an effectiveness for a number of months, and then you're going to have to get it again and again and again. Now, the advantage of the again, again is if there are side effects, if there are problems that you need to adjust the dose, you can adjust the dose. For a virus, because it is a one-shot treatment, it's harder to bake in that level of control to modulate if you need to tweak things. So we're, again, when thinking about different therapeutic approaches, everything has its pros and cons, but the virus approach could be really powerful if this works. And it's being currently tested right now for Rett syndrome, for example, another single gene form of syndromic autism that actually looks a lot like MCHS.
There has been a lot of people in the community who have given their kids either 5-MTHF, which is a over-the-counter available form of folic acid, or in some cases been prescribed leucovorin, and have seen really good results as far as both expressive and receptive communication. Leo is among those. He's on 5-MTHF, and it's subtle, but it's really noticeable if you
For
do him
us.
well. Yeah. So, do you know anything about MCHS and how it might relate to folic acid and that treatment?
Not really, to be totally
Okay.
honest. I do think that there are some of the peripheral immune
Mhm.
functions that MEF2C seems to play an important role in, that lipid signaling and lipid cascades and things like this are really important. Some things like folic acid are going to be foundational building blocks for several, important things happening in our bodies. So, and I think it's interesting as a family group. as long as, I would obviously always do this in consultation with your doctor, but trying things to see if it might have an effect is really, it's all you can do at this point. And
Right.
I think one of the exciting things about the fact that the family group is so connected now is that you can share your stories. You're not just an
Mhm.
N of one, as we like to say, out in the wilderness, trying to reinvent the wheel over and over again. And I think being able to share your experiences and your own little personal experiments, right? This is what you're doing when you're trying these kinds of things. It's really powerful. And I think it'll be interesting to see if something you stumble on something like this that has real effectiveness for, even if it's just a subset of you.
So, what are the next step forward, and what can families do to help?
I think the next steps forward for the team, for the MEF2C therapeutic team, is really trying to push these things forward in parallel, right? We're trying to test the TSB. We're trying to identify one that looks optimal. We're in the process now of trying to see if we can identify some potential industry partners to help us move this forward more aggressively. We're also working, again, very aggressively on the viral based strategy as well. And in addition, we're even beginning to try some additional things. So, for example, there are some FDA approved, there's a whole library of FDA approved compounds for treating this, that, and the other thing. We're actually now trying to see if we could design using human cells from maybe patient derived cells that we can convert into neurons in a dish and screening those libraries of FDA approved compounds to see if any of them increase MEF2C function, which then could potentially be prescribed off label to see if it could have some effectiveness in the kids or in individuals with MCHS. The other thing that's going on right now that you know of is really the patient registry, right? We're really trying to be sure that the family groups are lined up, that the symptoms are well documented, that we have a good understanding of what we're trying to improve. Because anytime you try to get a drug or a therapeutic approved through the FDA or other approving bodies across the world, you need to be able to say, these are the things we're trying to improve, right? So in a clinical trial, having clinical readiness data will be very, very helpful, not only to the family group, because we'll know who you are, we'll know where you are, we'll know who's eligible and who's not for clinical trials based on other factors, but also it makes therapeutic more attractive to a potential industry partner because they know what the patient population is. They know that we're ready for potential clinical trials, for clinical testing. So, I think for the family group, the thing that's really important is to stay active and engaged, remain hopeful, participate. As much as you can, try to participate in some of the clinical readiness types of studies that are going on. I know of the one in Cornell for sure. I think there's another in Boston. Being actively engaged in that is going to be really important for wherever we go with this in this journey of therapeutics. And it may be something else that comes up, other investigators out there developing similar types of therapies and ideas, and we just don't know what's going to emerge as the most effective thing. So, there's I think tremendous reason to be hopeful, but these therapeutic journeys have a lot of twists and turns and they take time for obvious reasons, right? We can't just, throw things in a human being and see what happens. So there's a lot of background work that goes into developing these. The other thing to bear in mind is that, as you all know, right, it's expensive to do this stuff, right? It's expensive to do the clinical studies. It's expensive to do the clinical readiness trials, clinical readiness, natural history studies, and it's expensive to do the therapeutic development projects. And so we're trying to leverage all the resources we can, right? We're trying to reach out to philanthropy groups that are looking to help rare disease communities like yours to support and fund things. We're trying to partner with government agencies like the NIH that has resources for helping to develop treatments and therapeutics for this type of thing. And then obviously working with just broad donors, et cetera.
Great. All right. Well, we really appreciate all your time, Dr. Cowan. Excuse me, I almost messed it up there. And as usual, our audience can contact us on Facebook or email us at mef2ccast@gmail.com if they have any questions. So thanks everybody for listening. And thank you again, Dr. Cowan.
Thank you. Enjoy the time.